Hello, my geniuses. Welcome to another biochemical lecture series today. It's a great science tutor, Lorenzo, and today we'll be doing the urea cycle. I get it now, the urea cycle. So we have done on transamination, we have done on deamination. So we're talking about the urea cycle here. So if you are new to my channel, make sure to subscribe and turn on your post notification button so at any time I drop any video, you'll be sure to be notified. If you want me to make a video for you, drop it down in the comment section. I promise I'll make that video for you. If you are confused on any part, tell me, put it down in the comment section. I promise I will, when I see it, I'll make that correction and maybe make a subsequent video that you use to understand what I thought. You get it now? So let us jump right into the video. So now if we are introducing what the urea cycle is, very simple. It is a metabolic pathway. Are you getting that? It is a metabolic pathway which involves the elimination of ammonia as urea. Very simple. Are you getting that? Now you are getting ammonia doing what? Oxidative deamination. Because first of all, your body has to break down your amino acid through transamination, and when your body transaminates by breaking down amino acid, what happens? It now undergo what we call oxidative deamination, and in oxidative deamination you remove ammonia. And when your body remove ammonia, your ammonia will be released out of the body as what? Urea. So you are removing it as urea. So the process of urea formation is called urea cycle. You get it now? So first of all, we have catabolism of ammonia. In catabolism of ammonia is through transamination, where you break down ammonia. You get it now? Remember, when your body during fed state, when you are feeding, you are increasing your glucose level, but during fasting starvation your glucose level drops. Your body now depends on other carbohydrate sources like what? Amino acid, fats. Are you getting now? So now during transamination, this is when you are transferring what? Amino group. That is just a transfer. But anytime you remove amino group, you are removing amino group in form of ammonia. And when you remove it in form of ammonia, it can either be stored as glutamine or it can be removed from the body. If you store it in form of glutamine, you want to use the glutamine to make what? Something else like pyrimidine. Are you getting now? You are using it to form pyrimidine or you are using it to form purine. Are you getting that? These are nucleotides. When you are storing your ammonia in form of what? Glutamine. So glutamate removes ammonia during what? Oxidative deamination, when it is converting from glutamate to alpha ketoglutarate. So when you are converting from glutamate to alpha ketoglutarate, what happens? You are removing ammonia, and that ammonia can either be stored as glutamine or you can remove that ammonia from the body as what? Urea. So the process whereby we catabolize what? Amino acid. Are you getting now? By removing amino group to form a keto acid, which is alpha ketoglutarate, thereby now removing the ammonia as in form of urea. We call it the urea cycle. So where does it occur? It occurs exclusively in the liver. Again, now it occurs there in the liver 100%. But now in the liver, now it has a mitochondrial part and it has a cytosol part. Not all of them occur in the mitochondria, not all of them occur in the cytosol. You get it now? The first two reactions, whereby formation of carbamoyl phosphate and formation of citrulline, occurs in the mitochondria, while in the cytosol you are forming the rest. Again, so it occurs in the mitochondria and the cytosol of the liver. So the next question you ask yourself, what stimulates the reaction to occur? So the first thing here is that when there's high protein diet, when there's high protein level, your body has high protein level. High protein level in your body can trigger what? Urea cycle, because you want the protein to be removed. You get it now? So if not, when you have high amount of protein, you have what they call hyperproteinemia, and hyperproteinemia can pose a clinical issue to you. So when you have high protein diet, what happens? Your body start inducing the formation of what? Urea, by removing your what? Ammonia. Also, are you getting here? There will be increase in amino acid catabolism. I told you high protein diet triggers the body to start breaking down the excess amino acid. Also during starvation and strenuous exercise, you can also have what? Urea cycle, and also there will be release of glucagon at that particular phase. Glucagon always released during fasting, starvation, strenuous exercise, and also your N-acetyl glutamate too can induce urea cycle. So what is the inhibiting factors now? In presence of liver disease, what happens? Metabolism reduces. In the presence of liver disease, metabolism reduces, so there's no urea cycle. Also deficiency, when there's deficiency of any of the urea cycle enzymes. So when the urea cycle enzymes are deficient, what happens? Urea cycle will not occur. Also, when you don't have N-acetyl glutamate, which is the allosteric activator of what urea cycle, when it is absent, there's no urea cycle. Also, there's what we call low protein diet. Are you getting that? So when you have low protein intake, your body will rather store the protein rather than breaking down the protein. And the last one is maybe you have any hyperammonemic disorder. Any hyperammonemic disorder will not trigger it to occur. So why do we have urea cycle? One, to convert excess ammonia into urea for safer excretion. So any ammonia in the body that is high, your body remove it by urea cycle. If not, if it's too toxic during passing, what happens? It can damage the kidney. So your body have to safely remove it by forming urea. Also, to maintain the blood ammonia level. So ammonia is also good in the blood because it helps to balance the body's pH. Are you getting me now? So, but you don't want excess ammonia because excess ammonia can induce cerebral edema. It can cause mental retardation. It can cause a lot of things in the body. It can cause imbalance of acid base in the body. Also, it is essential for nitrogen balance and survival. So nitrogen is needed in the body, but when there's excess nitrogen in the body, it causes issues. So now the next one is the biochemical pathway. We have talked about the introduction, we talked about where it occurs. Are you getting me? We talk about the stimulating and inhibiting factor, and the next one we are talking about is the biochemical pathway. In the biochemical pathway, it occurs in two places, in the mitochondria and in the cytosol. So you are looking at the pathway. So now watch me now. Here now you are condensing that ammonia that is released during oxidative deamination. There's an ammonia that is released there. The ammonia was released during oxidative deamination of glutamate. So the amino group in glutamate is removed as ammonia. So when it is removed, it forms alpha keto glutarate. That amino group that is removed in form of ammonia is channeled to the mitochondria to undergo urea cycle. So that ammonia there now that is removed as an amino group condenses with CO2, which is bicarbonate, in the presence of two molecules of ATP to form carbamoyl phosphate. So ammonia that was brought out as an amino group during the oxidative deamination of glutamate to form alpha ketoglutarate, by glutamate dehydrogenase is converted is condensed with CO2 in the presence of two molecules of ATP to form carbamoyl phosphate. By the enzyme carbamoyl phosphate synthetase 1. Okay, now so carbamoyl phosphate synthetase 1 catalyzes the condensation of the ammonia plus bicarbonate in presence of two molecules of ATP to form carbamoyl phosphate. So carbamoyl phosphate synthetase 1 is a rate limiting enzyme in the urea cycle, which helps in the short-term regulation, or you can call short-term or the fine regulation of urea cycle. In step 2, now what happens? Ornithine from the cytosol enters into the mitochondria and condenses with carbamoyl phosphate. So if it condenses with carbamoyl phosphate, it will form citrulline. So citrulline is formed by the condensation of ornithine. Ornithine is a basic amino acid that leaves the cytosol into the mitochondria, where it condenses with carbamoyl phosphate. So ornithine is where cytosol leaves the cytosol into the mitochondria, where it condenses with carbamoyl phosphate to form citrulline by the enzyme ornithine transcarbamylase. So ornithine transcarbamylase catalyzes the condensation of ornithine and carbamoyl phosphate to form citrulline. So citrulline now does what? Citrulline leaves the mitochondria into the cytosol. Ornithine enters the mitochondria to condense with carbamoyl phosphate, but citrulline that is formed now leaves the mitochondria. It leaves the mitochondria into the cytosol, where it condenses with aspartate. So citrulline leaves the mitochondria, where it condenses with aspartate in 1 molecules of ATP. So 1 molecules of ATP is needed to be broken down with ADP. So we need ATP here. So ATP is needed for the condensation of aspartate with citrulline to form argininosuccinate. So argininosuccinate was formed by the condensation of citrulline from the mitochondria and also aspartate in the presence of 1 molecules of ATP. So what enzyme that is catalyzing argininosuccinate synthetase? So if it's a synthesis reaction, what happens? We are breaking down 1 molecule of ATP. Now inside the same cytosol, what happens? Our argininosuccinate is lyased. Are you getting now? So our argininosuccinate is cleaved by what we call argininosuccinate lyase to form what? Arginine, and give off what? Fumarate. So argininosuccinate is cleaved by lyase enzyme. What is the lyase? Argininosuccinate lyase. So it's a cleavage reaction here, whereby argininosuccinate is cleaved by argininosuccinate lyase to give up what? Arginine and fumarate. Because argininosuccinate was formed by the condensation of what? Citrulline and aspartate in the presence of one molecule of ATP to give argininosuccinate by the enzyme argininosuccinate synthetase. So that argininosuccinate synthetase in the same cytosol what is cleaved by a lyase enzyme to form what? Arginine and fumarate. So fumarate does what? Enters into the TCA cycle. Are you getting me now? Where it gives what? Energy. So it can be used to form energy like that inside the electron transport chain. Now that arginine, now do you know what to do? Arginine is hydrolyzed by what we call by the enzyme arginase. Arginine is hydrolyzed by the enzyme arginase to form what? Ornithine, and giving off urea. Are you getting me now? So arginine is hydrolyzed by the enzyme arginase to form what? Ornithine. When it is forming ornithine, it gives out urea as a byproduct. So urea is given off anytime there's hydrolyzation of what? Arginine by the enzyme arginase to form what? Ornithine. So ornithine was formed as an end product of hydrolyzation of arginine. So when you hydrolyze arginine, you form ornithine, but at the same time you are giving out what? Urea. So urea is transported to the kidney for what? Elimination. Why ornithine is now used to do the next reaction, whereby ornithine now enters inside the mitochondria where it is condensed with carbamoyl phosphate for the cycle to what? Continue. You get it now? Very, very easy. So when you remember what we call CCAAO, CCAAO, C is the carbamoyl phosphate, the first C in the mitochondria. Second C is citrulline, both of them. So two C's are produced in the mitochondria. AA, which is argininosuccinate, arginine, and ornithine, they are produced in the what? Cytoplasm. The regulation of urea cycle now. What is the regulation? You can regulate urea cycle in short term and long term. So for you to regulate urea cycle in short term, we call it what we call fine regulation. If you are regulating it for long term, we call it coarse regulation. Short-term regulation now is using the rate limiting enzyme CPS1, which is carbamoyl phosphate synthetase 1. Carbamoyl phosphate synthetase 1 is regulated and allosterically regulated or activated by what? N-acetylglutamate. So I told you that the rate limiting enzyme is CPS1. Are you getting me now? But we also have another CPS2. CPS2 is important in what? Pyrimidine metabolism. Are you getting me now? So when you condense glutamine and what? Bicarbonate in presence of ATP, are you getting me now? You are forming using CPS2, which we are forming carbamoyl. So we can form carbamoyl phosphate in two places, in what? Urea cycle and in what? Pyrimidine metabolism. In urea cycle, ammonia is condensing with what? CO2 in the presence of ATP to form what? Carbamoyl phosphate. While in pyrimidine metabolism, remember I said we can store our ammonia in form of glutamine. So ammonia now in form of glutamine condensing with what? CO2 to form what? Carbamoyl phosphate. But that one is CPS2. Are you getting me? That one is CPS2, and which is allosterically inhibited there and activated by a different component. But here CPS1 is allosterically activated by what? N-acetylglutamate. So N-acetylglutamate is formed by what? Acetyl-CoA and glutamate. So when you condense acetyl-CoA and glutamate, are you getting now? You are getting N-acetyl-glutamate. So remember, in short term or fine regulation, the rate limiting enzyme is what? CPS1, carbamoyl phosphate synthetase 1, which is allosterically regulated by what? N-acetylglutamate. In the long-term regulation, which is the coarse regulation, it is what? Regulated dietarily. Are you getting now? It's regulated by dietarily. So remember, when there's already high protein diet in the body, when there's high protein diet that is stored, the body will start breaking the diet, which The body will start breaking the protein during fasting, starvation, strenuous exercise by the release of what? Glucagon. So already the body has stored protein during feeding, and when the protein level is high, which means the amino acid level is high, and if the amino acid level is high, the body during fasting, starvation, strenuous exercise will start breaking down the amino acid. When you start breaking down the amino acid, there will be transamination. From transamination, there will be oxidative deamination, and when there's oxidative deamination, your body will now start what? Undergoing urea cycle, so that I can remove all the what? Amino group in form of ammonia. Because when it is toxic to the body, it start producing what? Different symptoms called hyperammonemia. Okay, now so you can regulate it like that. You can regulate it dietarily. I get it. In long term, and you can regulate it hormonally. In the hormonal regulation, I told you that during fasting, starvation, strenuous exercise, the body releases what? Glucagon. Anytime that the protein level is high and the body is supposed to maintain the normal level of protein, the body secretes glucagon. So glucagon helps in what? Breakage and the carboxylation of the amino acid. So when it breaks down the amino acid, what will the amino acid do? The amino acid will be used to form ATP through alpha ketoglutarate by oxidative deamination. You get it now? Because if you go to my transamination video, it is the transfer of amino acid. Are you getting it now? So when you are transferring, it is the transfer of amino group. So after transporting amino group, what is the next one? You now start what? Removing the amino group in form of what? Ammonia. So the goal of transamination is to form glutamate. Are you getting it now? The goal of transamination, we are forming glutamate as the end product. You get it now? So why? Because we are using what? Alpha ketoglutarate. So when we form glutamate will now undergo oxidative deamination to form alpha ketoglutarate, which can go and form ATP. The glutamate that was oxidatively deaminated remove what? Amino group in form of what? Ammonia. So it remove ammonia, but first of all, it did not remove ammonia, it's removing the amino group, and as you are removing the amino group, that amino group is interpreted like what? Ammonia. So I can use either store the what? Ammonia in form of glutamine, or I do what? I remove the ammonia when the level is high. So if I'm removing the ammonia, what happens? I'm removing in what? Urea cycle. So glucagon is very, very important. Cortisol is needed here, and catecholamines are needed during the regulation, the long-term regulation of what? Urea cycle. The next one is the what? Energy requirement. So what is the energy requirement? Remember, in step 1, when I want to condense ammonia with what? CO2, what do? I use 2 molecules of glucose. So you are reducing 2 molecules of glucose there. So 2 molecules of glucose is lost at that particular point. And also, when we want to form argininosuccinate, remember argininosuccinate is formed by the condensation of citrulline and what? Aspartate. So when you are condensing citrulline and aspartate in the presence of argininosuccinate synthesis, what happens? You are using what? More molecules of what? ATP. So now 2 molecules in step 1, 1 molecule in step 3. I get it now. So we are losing a total of what? 3 ATP. 3 molecules of ATP is utilized during urea cycle, because 1 ATP is converted to what? AMP and what? 2 pyrophosphate. Therefore, the cycle consumes a total of 4 high energy phosphate what? Bond for synthesizing 1 molecule of urea. 3 ATP is lost, but due to the fact that ATP is always converted to AMP and pyrophosphate, what happens? So total, if you are calculating, total of 4 ATP, total of 4 high energy phosphate bond is what? Given out. I hope it all makes sense to you and you have understood everything I've talked about in the urea cycle. Remember, what is the essence of urea cycle? Urea cycle is important because our body is eliminating toxic ammonia in form of urea. So urea is a better form to remove what? Amino acid. Because if there's excess amino acid in the body, it will cause high protein. And when there's high protein, it can be dangerous to us. So we are removing it in form of urea. In urea cycle, it can occur in the cytoplasm and in the mitochondria. So remember, CCAAO. The first one is carbamoyl phosphate, followed by citrulline, followed by argininosuccinate, followed by arginine, and followed by ornithine. Are you getting now? And remember, it can be regulated by short term and in long term. So hope you have understood everything I've said and you understood it very well. So if you are new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. If you want me to make a video for you, drop it down in the comment section and I promise I'll make that video for you. So now we'll be going into another one, which are special amino acids. They are special amino acids. You get it now. So I'll see you in my next video. Bye bye.